Hi, my name is Alina, and today I'd like to present to you my project, Unveiling the Connection, the impact of SARS-CoV-2 infection and COVID-19 vaccination on Epstein-Barr virus reactivation. Let's start off with a quick intro. I'm currently a Bay Area student, class of 2024 in California, and my mentor is Dr. Kim Marie Dam. My research question asks about the relationship between severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2 infection and or COVID-19 vaccinations and their relation to the Epstein-Barr virus or EBV reactivation. The importance of this question is that more than about 81.4% of the world's population has received at least one dose of the COVID-19 vaccination, and more than 770 million people have gotten infected with SARS-CoV-2, making the possibility of either being related to the reactivation of EBV a major concern. You may ask, why is the reactivation of EBV a major concern in the first place? Well, this is because recent studies have shown that EBV is related to increased risk of multiple sclerosis, or MS, a debilitating disease because it may damage nerves in any part of the brain or spinal cord, therefore causing symptoms in many parts of your body. I'll talk about all of these in more detail later on. But first, let's start off with a quick intro to the immune system. White blood cells are cells part of the immune system that help the body fight infection and diseases. So there are many types of white blood cells, but today we'll be mainly focusing on the types of lymphocytes. What are T cells? T cells can kill infected host cells, activate other immune cells, produce cytokines, which are proteins that help control inflammation in the body and help control immune responses. NK cells destroy infected and disease cells like cancer cells. And third, B cells produce antibodies against antigens or foreign substances and can be targets in autoimmune diseases like MS. Antibodies are basically proteins produced by B cells that recognize and bind to specific antigens to neutralize or mark them for destruction by other immune cells. And plasma cells, which develop from B cells, are the ones that secrete antibodies. This process is the process in which plasma cells are developed from B cells is called plasma cell differentiation. A fundamental part of the immune response to injury or infection is inflammation. Pathogen detection triggers an inflammatory response, which involves the release of cytokines and chemokines, which are signaling molecules. These increase blood flow to the infected area, causing redness and heat, and make blood vessel walls more permeable, allowing immune cells to enter the tissue, leading to swelling and sometimes pain. Inflammation will be pretty important to the discussion of the links between these conditions and diseases later on. Chronic inflammation can contribute to the development of autoimmune diseases like MS. Autoimmune diseases are conditions arising from abnormal immune responses to a normal body part. And lastly, to go into a little bit more detail, we're going to talk about the angiotensin converting enzyme, or ACE2. What ACE2 does is help regulate blood pressure, which is really crucial for maintaining cardiovascular health. But why it's truly important in the context of all of this is that SARS-CoV-2 uses the ACE2 receptor to enter human cells. The spike protein of the virus binds to ACE2 on the surface of the cells, especially those in lungs, heart, kidney, and intestines. Before I get started, I'd like to note that since the end of the public health emergency, which started in January 2020, was on May 2023, data crucial to understanding the spread and impact of COVID-19 was reported by the government and other sources less frequently, or no longer reported at all. Therefore, some data may be a little outdated, but still accurate. So COVID-19 is an infectious disease caused by a strain of coronavirus called severe acute respiratory syndrome coronavirus 2. It's primarily contracted from airborne transmission, but can live on surfaces for 2 to 3 days, so therefore there's a rare chance of surface transmission. When SARS-CoV-2 enters your body, it latches spike surface proteins to receptors through the ACE2 receptors, and therefore that's how it enters the respiratory tract. Throughout the body, the ACE2 receptors mark the tissues vulnerable to infection by allowing viruses to enter the cells through it. And inside the cell, the virus takes control and replicates rapidly, invading other nearby cells. If the immune system doesn't get rid of and shed SARS-CoV-2 enough during this initial phase, the virus then turns to attack the lungs, where it can turn deadly. If infected with SARS-CoV-2, reactions may vary a lot appearing within 2 to 14 days of infection. So some common symptoms are on the screen right now. And over periods of time, mutations in SARS-CoV-2 have brought about infections of several variants of the virus, each with different abilities to affect both the vaccinated and unvaccinated populations. Here is a graph of a general trajectory of COVID-19 as of November 2023 by the World Health Organization. As you can see, COVID-19 first emerged in around 2019 with related deaths peaking in 2021 and cases peaking in January and December 2022, and then continuing to decrease until 2023. So many people completely recover within days or weeks of infection with SARS-CoV-2. However, almost 16% of them never seem to recover fully, retaining lingering symptoms for weeks, months, or even years after their initial diagnosis that may worsen with physical activity, etc. And this is called long COVID. And in those with long COVID, researchers have found T cell alterations. Basically, T cells are exhausted or reduced and at increased amounts of autoantibodies. Currently, studies found that there are likely multiple potential causes of long COVID, ranging from persisting amounts of SARS-CoV-2 in tissues to autoimmunity or immune dysregulation, with possible reactivation of underlying pathogens like those of EBV, possibly responsible for this. Because it is a physiological condition that can affect multiple body systems, some with long COVID undergo increased risks of cardiovascular disorders, in addition to other disorders such as mental health disorders. Additionally, some symptoms last for years or are even expected to be lifelong. The substantial loss of health due to long COVID underscores the importance of understanding the health effects from SARS-CoV-2 infection and EBV reactivation. Next, on to COVID-19 vaccination. So, although in most cases, COVID-19 vaccination does not lead to serious side effects, and more often than not protects you against COVID-19, more recent reports have suggested links between COVID-19 vaccination and EBV reactivation. Currently on the screen are a few examples of available COVID-19 vaccinations around the world. And as you can see, there are many different types of vaccines, from bivalent to mRNA to adjuvanted and recombinant. The last one is currently not available in the US due to the fact that it has expired, but it did exist before 2023. Now on to the kissing virus, or EBV. EBV is one of the 9 known human herpes viruses and infects more than 90% of people worldwide. It's the most common causative agent of infectious mononucleosis, or mono, which you may have heard of. It's a contagious disease that is characterized by swollen lymph glands, fever, sore throat, and extreme fatigue. As a herpes virus, EBV has both a productive or lytic and a non-productive or latent phase, as you can see on the graph on the slide currently, with productive infection in the oral mucosal epithelium and long-term latent infection in the body as well. So this latent infection persists for the lifetime of the host and is invisible to the immune response, but EBV may be reactivated by sickness in those with weaker immune systems. In rare cases, EBV may lead to chronic active EBV infection, or CIEBV, which is a serious condition where infection persists more than 6 months after initial monodiagnosis because the body's immune response cannot control the infection. Unfortunately, CAEBV can lead to death. Infection with EBV happens through the transfer of bodily fluids, especially saliva, therefore the kissing virus. Recent studies have found that in the endoplasmic reticulum, or the ER, the Xbox binding protein 1, or XBP1, which is essential in plasma cell differentiation, could activate EBV gene expression with the sigma-1 receptor, or S1R, a modulator of multiple signaling pathways. The binding of SARS-CoV-2 interacting proteins with S1R plays a significant role in promoting the viral replication cycle. Therefore, these receptors are ideal targets for designing antiviral therapies to curb viral replication. And accordingly, S1R agonists like fluvoxamine may help prevent EBV reactivation as it attenuates COVID-19 entry and may decrease COVID-19 replication and ER inflammation. There are currently no vaccines available for EBV due to it being a challenging target as it kind of changes along its life cycle and can cause tumors to develop. So it's currently out of the question due to possible increase of cancer. However, there are evaluated antiviral drugs such as ganciclovir that inhibit EBV in vitro. And the findings on S1R agonists represent the possibility of creating therapeutic drugs that limit EBV reactivation and long COVID symptoms. With the recent evidence on links between EBV, MS, and COVID-19, among other diseases, there are now 3 EBV vaccines in the making. Lastly, on to multiple sclerosis, or MS. It's the most common chronic neurodegenerative autoimmune disease, affecting 2.8 million people worldwide. Although its exact etiology remains unclear, it's considered a complex disease, with 3 main group factors involved in its pathogenesis, namely immune factors, environmental factors, and genetic associations. So MS is identified by demyelinating lesions in the brain. And what happens during this is that the protective covering or myelin sheath around the nerve fibers in your central nervous system are damaged. So this also happens with the infiltration of autoreactive lymphocytes into the central nervous system, which causes chronic inflammation. This following inflammation may cause the breakdown of the blood-brain barrier that shields us from bacteria that may be in the bloodstream. Consequently, individuals with MS may have affected vision, sensations, impaired motor function, and cognitive and emotional functions. Currently, there are no widespread public vaccines for MS, but current research reveals a promising potential vaccine developed by researchers at the University of Chicago, using a process much unlike the typical vaccine. Instead of helping the immune system recognize the virus as the enemy, it would completely reverse the autoimmune disease without shutting down the rest of the immune system by removing its memory of the molecule. And additionally, while currently there is no cure for MS, treatment is found with ongoing clinical trials that help reduce symptoms and slow the disease. So now that I've talked about all of these diseases and conditions, you may be asking, what's the relation between all of them? If you look at this graphic on the slide currently, studies have shown that SARS-CoV-2 infection and COVID-19 vaccination can both lead to the reactivation of EBV. Some COVID-19 mRNA vaccines have been associated with increased T-cell response, which activates macrophages and may contribute to B lymphocyte tumorigenesis and inflammation. Similarly to the effects of COVID-19 vaccination, researchers have found that infection or co-infection with SARS-CoV-2 can change the immune cell response. So there was production of less spike-specific, lower functionality T cells, especially if the people weren't vaccinated. So SARS-CoV-2 infection therefore tends to produce an immunosuppressive state with functionally impaired and decreased T lymphocytes, exactly what can trigger EBV as it is an opportunistic virus. Usually, infection of EBV leads to asymptomatic or mild cold-like symptoms and reactions. However, recent research has found that EBV is related to a wide range of more serious illnesses, including around 200,000 cancers per year, and also autoimmune disorders, the most notable being MS. The risk of MS increases just about 32-fold with EBV infection, and even more with symptomatic to severe mono, as latent infection is a source of viral stimulation, and multiple EBV antigens are the targets of cross-reactive autoantibodies found in MS. As a key characteristic of MS is chronic inflammation, and EBV-positive lymphocytes may exacerbate that inflammation in addition to SARS-CoV-2 infection and vaccination, causing a strong inflammatory response in the immune cells. Continued inflammation after viral clearance and evidence of atypical cytokine activation in patients may contribute to long COVID. Accordingly, Determining ways to minimize the mechanisms that cause inflammation may help relieve some symptoms for those infected with long COVID, MS, EBV, COVID, and COVID-19. So with increasing widespread links between EBV and MS, and studies that suggest associations between EBV, COVID-19 vaccination, and SARS-CoV-2 infection, this field of study has proven itself an urgent need of further research. This research reflects the importance of an understanding of possible factors that may influence a COVID-19 vaccination, leading to further disease, such as the health history of patients with SARS-CoV-2 infection. Currently, there are several possible treatments aiming to relieve symptoms of individuals with EBV, MS, COVID-19, and long COVID, and vaccines aiming to prevent EBV are additionally being developed and evaluated. Although increasing evidence points to a relationship between EBV and COVID-19, currently none are strong enough to reach a definite answer. Therefore, this link also needs to be addressed in order to gain a better understanding of minimizing EBV reactivation and decreasing the likelihood of MS and long COVID symptoms. Thank you for listening. I would like to express my thank you to my brilliant mentor, Kim Marie Dam, as well as Luke Fisher and Lalita Lemay for helping me with publishing and revisions. Thank you for listening.